Forget frequently asked questions.
Common sense, common knowledge, or Google.
How about advice from a real genius? 95% of people in any profession are good enough to be qualified and licensed. 5%
go above and beyond.
They become very good at what they do, but only
0.1%
are real geniuses. Richard Jacobs has made it his life's mission to find them for you. He hunts down and interviews geniuses in every field: sleep science, cancer, stem cells, ketogenic diets, and more. Here come the geniuses. This is the Finding Genius Podcast
with Richard Jacobs.
Before we begin, a note from our sponsor. I'm Richard Jacobs, Executive Director of the nonprofit Finding Genius Foundation and host of the Finding Genius Podcast. In late 2016, I was rear-ended at 65 miles an hour by a truck on the highway, which sent me off-road into a ditch. The impact of the collision gave me a concussion and other injuries. At the hospital, a CT scan showed that I had thyroid nodules, which turned out to be cancer. It was then, when I had a biopsy of my neck, that I realized even if I was a millionaire, I wouldn't want a second or a third biopsy due to the pain and the invasiveness of it. And appointments at that time for thyroid experts were 3 to 6 months out. And I was worried about dying now, even if that was irrational. So because of this, I've decided to raise money to conduct a literature review on steroids, on the causes of anxiety and depression, a condition that affects well over 50 million people in the United States and hundreds of millions worldwide. Our goal is to create a codex, a guide that reveals all possible treatments for anxiety and depression for people that live with the condition, or for loved ones that have it, as my wife and my son do. To find out more about our fundraiser, visit findinggeniusfoundation.org and click on current initiatives. And now, to our guest.
Hello, this is Richard Jacobs with the Finding Genius Podcast, now part of the Finding Genius Foundation. I have Dr. Eric Bapteste. He's a CNRS researcher, all part of the Université of Pierre at Marie Curie University in France. I'm going to talk about evolutionary biology and his research. So, Eric, thank you for coming.
Thank you very much.
If you would, tell me about your research.
Sure. So what do? We're interested in the evolution of interactions. We care for what biology cares about, that is interaction, but from an historical evolutionary perspective. So we want to understand how different kind of entities, could be molecules, could be organisms, interact with one another, and what are the outcomes of these interactions. The general idea of our work is to try to track the evolution of processes and eventually the evolution of ecosystems rather than the evolution of species.
So what are some models of interaction that you've been looking at so far?
Well, we are first describing what we think are the interactions taking place in nature. So for this, we basically register what is present with what frequency, with what numbers. And that gives us a backbone that you could see as an ecological description of the world or a systemic description of the world. And this backbone is basically a network of interaction. When we are interested in a given question, we want to know what is present, what is interacting with what. And given that backbone, then we want to see how dynamically it evolves over time. So the first model we need to use is a network of interaction. It could be interaction between genes, between proteins, between organisms, between biotic and abiotic components. But once we have this first structure, then we can try to ask questions. We can analyze the graphs using network sciences and using concept of evolutionary biology.
But what's an example of a system you've already used and analyzed, and what did you discover?
Good question. So, for example, that's more of a prospective discovery than of a discovery, really. But you know that viruses are genetically interfering with their host. Viruses need host cells to replicate. And when they fulfill their life cycle, viruses express their own genes, and they also manipulate, hijack the cellular functions. So what you have in effect is an interaction between genes, proteins of viral origins with genes, proteins that are coming from the host. And that creates a network that is interesting because it is composed of components coming from different sources. And the question you can have about this is what host processes are being hijacked, manipulated by viruses. So typically this is interesting because when you start connecting the viral proteins or the viral genes with that of their host, you do realize that maybe viruses are connected to very central genes or very central proteins of the host. If you have a virus that is pirating genes or owns gene copies or owns regulators of genes that are very important for the host, then you will see that with a network because you will have your viral genes or your viral protein connected to a very central, very important host node. That's an example of how one can use network, and the kind of discovery you will make then is about what processes in the host are manipulated, hijacked by viruses, for example.
So you're looking at it just more of an overview, of a systems view, you mean, instead of a single gene or a single protein?
You're right. There is a big holistic viewpoint here because you could look at any kind of interaction, truly, as long as it is solid, trusted interaction. You don't have to have a privileged gene to focus about, but you can also care for pretty specific systems. You can track, for example, specific domains within genes. Genes are composed of a smaller component, and these smaller components, some of them have received names. They're called domains because they correspond to functional units that are together building up genes. And so you can decide that you want to know for a very specific genetic domain with how many other genetic domains it is associated all over the protein universe. And if you do that, basically you can start from a very specific domain and look for its neighbors in a network that would show these associations. So you can have it both ways. You can start from your favorite gene, or you can have a broad view of what is interacting with what. And the question for us is, how did this interaction evolve? Where do they come to be? How did different partners integrated with one another in a functional way?
Well, how do you think viruses have adapted to target specific cells in certain creatures? How do you think that happened?
Well, that happens because viruses, in some sense, have their own selfish interest. And one of their interests is to replicate manipulating the host. For example, if you have a host like us with defense systems, it is important for the virus to fight these defense systems. This would be called a harm race. This would be called coevolution in some sense. And because viruses need to overcome our defenses to replicate, then if it can do it by modifying, affecting, regulating how we genetically regulate our own defense systems, and the virus that are able to do this will be selected for, positively selected for. Evolution will favor the one that are able to manipulate the host to weaken the host defense systems. And this is, we believe, how it can happen. Basically, it's a process of selection that eventually will favor some interactions over others.
Are you trying to model selection itself, or how do you even model this or get any useful information out of it?
No, it's a good question. We are trying to do things in a very simple way. Selection is often classically in population genetics modeled by basically counting how many offspring a given entity is going to produce. And so fitness is often equated with reproductive success. For interactions, it's more difficult because you can have interactions between a different kind of entities that are not per se reproducing. And therefore, what we are trying to look into is within the structures of the network, whether there are some interactions that are over time occurring again and again, that are robust to change, or that are resilient to change, meaning that if they go away, they come back, and eventually with frequencies increasing in the population of described interactions. So this is almost like a topological search. You take a network, you give weights to the edges composing the network, to the nodes that are present, and you search for nodes that are increasingly abundant, edges that become increasingly strong, for example. And if you observe this kind of pattern, and this is in a series of network over time, then you have a good hint that something right is done in a sense by this interaction and those interacting partners, because they manage to thrive in more abundant, stronger proportions than they used to before.
Are you trying to predict then interactions? Are you trying to predict outcomes? I mean, what's the modeling?
That would be nice, really, because what I'm describing is an attempt from an evolutionary biology evolutionary biologists to basically move from a description of what is related to what, which is classic phylogenetic perspective, to modeling, in a sense, the evolution of ecosystems, that is the interaction between different species, not just what is related to what, but what functionally works when different species are interacting together in an environment.
Before we continue, I've been personally funding the Finding Genius podcast for 4.5 years now, which has led to 2,700 plus interviews of clinicians, researchers, scientists, CEOs, and other amazing people who are working to advance science and improve our lives and our world. Even though this podcast gets 100,000 plus downloads a month, we need your help to reach hundreds of thousands more worldwide. Please visit findinggeniuspodcast.com and click on Support Us. We have 3 levels of membership from $10 to $49 a month, including perks such as the ability to see ahead in our interview calendar and ask questions of upcoming guests, transcripts of podcasts you're interested in, the ability to request specific topics or guests, and more. Visit findinggeniuspodcast.com and click Support Us today. Now back to the show.
And so in terms of prediction, what could be especially important would be to know whether a given ecosystem is stable, meaning it is composed of robust or resilient interactions, or whether this ecosystem is about to tip, or whether this ecosystem is in fact somehow declining. And so, yes, you need to be able to find a way not only to model the interaction in your ecosystem of interest over time, but also to try to capture the network features that makes you believe that this is a network that's going to be stable for longer, robust, or in fact that it is a network that has weaker parts, weaker interactions, weaker components, and would be sensitive to changes, random changes, catastrophic effects of various kinds. And so prediction is one of the desirable outcomes. It's not something we know how to do yet, but knowing that we are now in the Anthropocene and the environment is threatened, it is indeed a good idea to be able to predict whether the evolution of the environment is looking good or is going to be tragic.
So, how are you supposed to evaluate these systems you're looking at? Are you just looking at, in viruses? When you get infected by a virus, from what I understand, you're not infected only by one exact viral sequence, but the viruses will have different sequences slightly. So, can you evaluate reproductive fitness there? Which viruses and which sequences are successfully interacting with cells and which are not?
Right.
Or how can you model what you're talking about?
Okay, no, so that's not exactly what we are doing, but that is exactly part of what we should be doing, because what we want to know is how different entities, different viruses with different sequences are going to succeed or not succeed in a given system to multiply, and whether this ultimately is going to lead the system to crash or eventually is compatible with persistence of this collective composed of infecting viruses and the host population. So the way you want to model this is basically by integrating knowledge from different sources, from different observations. What is especially important is that networks are very flexible and they allow you to basically put inside a given framework knowledge from microbiology, knowledge from biology, knowledge from sequence evolution, and also, I would like to say, medical knowledge in a sense, because if you know that a given population or a given ecosystem, based on what a medical expert or say if this is the ocean, an oceanographer would say about its health, then you can integrate this information. It's called metadata, and you will see, that's an ecosystem that is doing bad, and this is how it is structured. It is basically maybe under attack by a very nasty invader, a very mean virus with a given specific sequence. Or if you have another medical viewpoint, you can say this is actually a situation where the host can handle the multiple virus attacks. And so you need a qualifier that's going to be provided by someone whose expertise is to tell you this is an ecosystem doing well, this is a host population doing well. And that's important, again, because network is about integrating different scientific practices.
So is there a particular network that you're using right now to study? And what have you learned from it? Or is this more early days and you haven't gotten there yet?
Yeah, it's in many sense, it's early days, and that's why it is very exciting. And also in many sense, we're studying a lot of such networks. And the reason why we can study many of them is because there are plenty of interaction networks that are well documented now, publicly available.
If you like this podcast, please click the link in the description to subscribe and review us on iTunes.
So if you see protein-protein interaction networks, you know that some proteins are associated with aging, for example, in humans. And so you have a big network that tells you what proteins known to be associated with aging is connected with what other proteins. And that allows you to make some predictions about the functions of the neighbors of proteins of interest. So, for example, protein A is associated with aging, but it is connected to protein B that is with another connection also connected to protein C that is also associated with aging. So maybe B in between A and C, both associated with aging, maybe B is also it hasn't been demonstrated yet, associated with aging. This is called guild by association. And so what we are doing very specifically in my lab, we are searching by guild by association what genes and what proteins, be they carried on viruses or be they hosted in the host genomes, may be associated with aging based on the interaction we know between proteins and genes already demonstrated to be associated with aging.
So you're looking for the presence of, let's say, certain genes or proteins in an aging process that are with known proteins and genes that cause aging. So you're trying to figure out what these ancillary ones that are also participating, or what do you mean?
So what we're doing is exactly this. We're mining networks to identify, based on neighborhoods within a given network, what are the likely functions of the genes that are also present in the same region of the graph. So if you imagine a network with colored nodes, and the nodes represent proteins that are, say, associated with aging, and you put them in red for some reason, then you will notice many regions that are full of red. And in between the red nodes, you will see densely connected with them some non-red nodes. Let's say make them white. And you will wonder, why is this white node connected to so many red nodes? Is this statistically significant? Is it expected? Does this suggest that the white node should be red? And this is how it works. So we're doing that, and we're in particular looking into genomes of viruses, whether they carry some homologs, copies of the genes that are white in my aging protein network and connected to lots of red proteins. So basically, are the viruses regulators of human aging? Is it the case that the genes they carry are going to be connected with the genes associated in aging and the protein associated in aging in human networks? That's an example of an application we're doing in the lab.
Are you saying that certain viruses are connected with aging? Has that been established at all?
That's what I'm saying, yes. And
so these would be long-term ones that are latent within us or active for many decades, or when or where would they appear?
It could be both, because basically there could be viruses that are happy to accelerate your cellular aging because they don't need your cell as much as your body needs them to fulfill their replication cycles. The viruses may be happy to kill your cell faster, get them to become senescent, or it could be latent viruses. But in the case of latent viruses, the other exciting possibility is that they do not manipulate aging. So we could actually use the latent viruses to later in the future, as a Trojan horse, integrate some additional genes that may manipulate aging. That has been tried experimentally by scientists trying to transform latent murine viruses into murine viruses carrying telomerases. And the idea was that modified viruses could interfere, and extend the lifespan of infected mice. And they showed, also the paper is currently only on bioRxiv, that it seems to be the case that when you change in a latent virus that is not interacting with your aging, some of its genes, so that it carries some genes that could do you good or do the mice good, then the mice live much longer, like 30 or 40% longer. So yes, in principle, it could be latent or non-latent viruses, and it has a different kind of interest depending whether it is a latent virus or not a latent virus. But this is a research question. We want to know to what extent viruses that are able to infect humans or able to infect the microbiomes of humans, it would be phages in this case, are potentially able to interfere with human aging? That's a research question.
Yeah, I was going to say, by definition, the viruses that would affect a mouse or a person, I mean, the only reason we know about them usually is the creature is sick, not doing better, not living longer.
Right.
But are you looking at phages, or are you looking at viruses that affect mice directly?
So we're not looking in mice, right? I was mentioning an experiment by a colleague scientist that you can find online, but we are looking with bioinformatic means in phages from the microbiome, in viruses that are known to infect humans, whether the gene content of these viruses and the gene content of these phages is comprised of genes that resemble, are similar, are homologous to the ones that in humans are associated with aging or to their regulators. So you see, it's
again. So you're looking at phages in people's microbiomes, or you're looking at viruses that make them sick to see what
both. Absolutely both. Yeah, and
I think it's a big enough job to just look at one. I mean,
no, it's not.
Microbiome science, I know, has been kind of restricted. Seems like mostly to the bacteria, but you're looking at the phage-ome then of people and seeing okay. Yep. So what have you identified? How many different phages?
I don't know. It's a research in progress. We'll know shortly. But the idea is that also those are very large data sets because precisely we're guided by the network structure. We know what to look for. We want to look for those genes that are either the direct neighbors of the genes of interest in our network, or that are themselves the red nodes I was mentioning about. So basically you start from the human sequences and you make a sequence comparison across all the genes that have been sequenced to date in phages and then in viruses. And do you find a match or you don't find a match? And because phages are not humans and because viruses are not human, you do not expect to see a match. But if you see a strong similarity between a sequence that is currently sitting in a virus or in a phage, then this is something astonishing. And you need to explain why this similarity.
Why are you skipping the bacteria? Why not look at the target of the phages, which is the bacteria, instead of our cells? I mean, even if the viruses have sequences that are similar to regulatory sequences in our DNA, who's to say that they're interacting directly with us and they're not really phages? They're you're supposed to be interacting with bacteria.
No, you're absolutely correct. And so then the question is about dealing with big data set, indeed, and finding the relevant ones. What is especially exciting for us are these what's called MAGs. So it's assembled metagenomes. So genomes from metagenomic data that have been reassembled to obtain what would be the highest quality, most complete genomes from bacteria living in us or on us. And so those are good data set to mine. You're absolutely correct.
So you're also looking at the bacteria as well and their genes and this whole chain?
We're doing what we can, and I'm going to make you a slightly deflationist answer here, but we are a small French team, so we do what we can.
So this current experiment you're running, when do you anticipate you're going to get your results? How far along is it?
Within months.
Okay. Do you have any preliminary results you can share, or you have to keep it all quiet until it's done?
Yes, nothing I want to share now today, but you were asking me for a specific example of how we were using networks for specific research questions. And I was telling you that in general, it was a good idea to study networks and the evolution of interactions because there is a lot to be learned about the stability of systems and the interaction between different kind of partners. And then I gave you a specific kind of example on a specific kind of research.
Well, has anyone even identified, all the phages that are typically associated with, any given person? I mean, the microbiome itself is, I guess it's hundreds of players.
Yeah.
And it's different in each person.
Yep.
Is the phage-ome on that scale, or is it thousands or tens of thousands, or how many?
Super big. I doubt this has been exhausted. And this is another thing we do in the lab. We are mining microbial dark matter. And to do this, we do this in a different way. We basically, there are many unculturable microbes and viruses, and we only know them through traces of their genomes, bits of their genes. And so that's called the microbial dark matter because it's a lot of genes from the environment. We basically don't know who is carrying them and what do they code for. So we're trying to search whether in this microbial dark matter, there may be hints of very divergent microbial groups that would belong to lineages that are very different to the currently known lineages. And therefore, this microbial dark matter, as you mentioned, is especially exciting because it has potential to unravel life as we do not know it. And you're right, we are far from having exhausted this microbial diversity. This is a big research theme in my lab. We want to know what's out there, and especially whether there are very surprising, shockingly divergent lineages in those microbiomes.
How many, are you taking microbiome samples from people? And is your cohort composed of X number of people? Or how many different systems are you looking at within people? How many different microbiome systems?
So we're not specifically looking within people for those ones. Because we're more specifically looking for environmental data sets. And the challenge here is to be able to look into the sequences of gene families that in general, across life forms and across microbes, are sufficiently well conserved and not too frequently laterally transferred, so that if you find a strange sequence belonging to this kind of gene family, you know you are looking at something weird. So basically, the first step is to identify gentle, kindly evolving gene families that are basically with the sequences allowing you to sort archaea on the one side, bacteria on the other side, and you would not confuse the bacteria for anarchy based on the sequences. And then, based on these gene families, you expand them with sequences that are similar to them in the environment, and with sequences that are similar to these similar sequences in the environment, and with sequences that are similar to these similar sequences in the environment. You do iterative sequence comparison, but at the same time, you make sure that when you make a long chain of similar sequences grow, they always align over the same initial starting region. So that means basically they are very divergent from the sequences you started from, but they would align over the same region, so they would be homologous. And when you grow gene families that are, in principle, as we know them, very kind and gentle in their evolution, and we notice that they are suddenly connected with sequences that are very divergent, very different, then it is exciting for us because we know that this gentle family is not so gentle in the environment, and then we have to understand why.
I just wonder how you're getting your data sets. Are there phage-ome data sets?
Publicly available data set. This is something very important, by the way. Again, we are a French team. We are not very rich. We are doing fundamental research, so we rely a lot on publicly available data sets. And typically, an example is a Evoloution data set. It's a good data set to mine, but there are plenty of other data sets. The most exciting data set for us nowadays would be the one with genomes assembled from the environment, so the MAGs I was telling you about earlier. But what we do in my lab is that we develop the methods to mine these data sets. We don't build the data set. We exploit the data sets that are available to us, and through collaboration, when people are interested and they want to know whether there are weird stuff in their environment, then they come to us and we have a look.
Well, very good. Eric, what's the best way for people to find out more about your research and to keep tabs on your upcoming papers?
I think it's, before COVID, it used to be by meeting people and getting in touch, around posters and talks and informally. And now I think we rely a lot on our pre-existing network of collaborators. And we hope that our good fame is going to bring us new collaborations. So it's a quiet, efficient process of trust between scientists. I think it's very important.
Okay, well, very good. Again, is there a website or a place people can go to learn about your work? Where should they go?
Honestly, the best way is to do a PubMed search on our work because this would cover more comprehensively the diversity of the research we do. We also have a lab website. I'm not very sure it is extremely up to date. It's www.evol-net.fr. So you could go there to have a glimpse of what we do.
Well, very good. Eric, thank you for coming on the podcast. And I know it's late evening in France, so thank you for being here.
Thank you, Richard.
If you like this podcast, please click the link in the description to subscribe and review us on iTunes.
You've been listening to the Finding Genius podcast with Richard Jacobs. If you like what you hear, be sure to review and subscribe to the Finding Genius podcast on iTunes or wherever you listen to podcasts. And want to be smarter than everybody else? Become a premium member at findinggeniuspodcast.com. This podcast is for information only. No advice of any kind is being given. Any action you take or don't take as a result of listening is your sole responsibility. Consult professionals when advice is needed.